Hello, I'm Chris Curran from Northern Kentucky University. This is the video version of my presentation at the 2013 Kentucky EPSCoR Annual Conference. The overall focus in our lab is on genetic susceptibility to widespread pollutants, and the primary focus is on polychlorinated biphenyls. There are 209 possible PCB congeners. Those with more chlorine atoms are resistant to degradation and bioaccumulate in the environment and in our food supply. Contamination has been reported worldwide, and this map shows the current advisories for PCB-contaminated fish in Kentucky. Children are at highest risk of adverse neurological effects from PCBs, so we are most interested in how PCBs affect the developing brain. Genetic differences can also affect susceptibility, so we use mice to model known human variation in two genes related to PCB metabolism, the aryl hydrocarbon receptor and CYP1A2. Coplanar PCBs bind to the AHR and move into the nucleus to increase expression of CYP1A2 protein, which is capable of sequestering PCBs and preventing their distribution to other target tissues such as the brain. Pregnant dams are treated once during gestation and once during lactation. Then their offspring are tested when they reach young adulthood. Our previous studies showed that CYP1A2 knockout mice are more susceptible to developmental PCB exposure than wild-type CYP1A2 mice when we tested spatial learning and memory using the Morris water maze. Our current work focuses on motor function because there are recent reports that indicate PCBs increase the risk of Parkinson's disease and other motor impairments. The brain regions of interest are the substantia nigra and striatum, which contain the major dopaminergic pathways affected in Parkinson's, and the cerebellum, which is required for motor coordination and balance. Our stereology system, funded by an EPSCoR equipment grant, allows us to look at gene expression in both of those brain regions. Here you see a normal substantia nigra and the cerebellum, where we are measuring expression of CYP1A2 and a related enzyme, CYP1A1. The rotarod test tells us if cerebellar function is normal. We found that PCB treatment significantly impaired rotarod function in our mice. Female mice showed greater impairments than males. We also found a genotype difference, with CYP1A2 knockout mice having significant impairments. This suggests CYP1A2 is required for normal cerebellar development or function. We used 3 tests to assess nigrostriatal function: the balance beam, the pole test, and gait analysis. After 2 days of training on the balance beam, mice are challenged with a wire mesh. We measure the time to cross the beam, the number of steps, and the number of slips per step. The pole test requires the mouse to turn 180 degrees and descend the pole back to its home cage. Non-toxic paint is used to mark the hind paws of the mice for gait analysis. The length and width of the strides are measured, as well as the difference between the shortest and longest stride. Our preliminary results showed no significant differences in these tests. Sticker removal tests sensory motor function, and we found a slight increase in the time required to remove the sticker in PCB-treated mice. We have shared these results at the National Society of Toxicology meeting, where Amy Ashworth and Anna Lang received undergraduate research awards. Austin Brown earned honors at the 2012 Kentucky Academy of Science meeting for his project on PCBs as obesogens and a risk factor for diabetes. We are continuing to explore PCB effects on metabolism because there is a significant weight difference in our animals as they age. We thank our funders from NSF EPSCoR, the National Institute of Environmental Health Sciences, and Northern Kentucky University for supporting all of this exciting work on gene-environment interactions, and, of course, our hardworking undergraduates. Thank you for your interest in our work.